Welcome back to this interdisciplinary course on organic chemistry in biology and drug design. Now, last time when we ended, we was discussing at that time the sequence determination of proteins. Sequence determination means the determination of the amino acids that are arranged one after another, and how to do that. We have just a recapitulation of that. That first it was discovered by Sanger, that how to know the N-terminus amino acid by using a reagent which is now popularly called Sanger's reagent, that is 2,4-dinitrofluorobenzene. And then a very similar reagent that was also introduced just to enhance the sensitivity of the determination, and that is the dancing chloride. And then for the other end, that means the C-terminus end, carboxypeptidase was used. That is an enzymatic method to know the C-terminus amino acid. While doing so, you can actually tell the last few amino acids, because they will come sequentially one after another. So, if you determine which amino acid is released at different time points, one is able to determine up to certain extent the first 3-4 amino acids. However, the determination of this complete sequence of amino acids in a protein or a peptide is what is called Edman degradation, where the reagent is phenyl isothiocyanate, and phenyl isothiocyanate has a beautiful chemistry evolved around it, and that is that after the attack by the N-terminus amino acid to phenyl isothiocyanate, which is an electrophile, and then that ultimately induces a cleavage of the first peptide bond, resulting in a peptide which is one amino acid less than the starting one, and you can isolate that peptide, and also you can identify which amino acid has reacted with phenyl isothiocyanate, and you can repeat the process. However, there is some limitations ultimately, and that is due to the not due to the fact that Edman degradation is the reactions are not very quantitative, the reactions are very high yielding, more than 90%. However, you have to remember that even if there are steps which are 90% yielding reactions, after the second step the reaction becomes 81% yield, and so it drastically reduces, because it goes every step. If we consider that is 90%, then after 10 steps it will be 9, the percentage is just to multiply 10 nines, that will give you the yield, and that will divided by, remember, divided by the by percent, it is a percentage. So, the percentage has to be there, ultimately the percentage will be very low. Just an example, suppose you do 4 steps. So, the amount will be 9 into 9 into 9 into 9. So, that will make it 81, then 72 and 63. So, finally, it will be about 63% yield, that is after 4. So, it drops down, and the limitation is currently that you can go up to possibly up to about 30 amino acids sequence per cycle, in one go, in one shot of Edman degradation. Okay. So, if you have a protein, so we ended up there that if you have a protein which is a very large, which is usually very large. Now, the question is what is the protein or difference between protein and the peptide. Sometimes we call peptide, sometimes we are calling proteins. Usually it is basically the molecular weight. There is no hard and fast rules, but if something contains less than 50 amino acids, that is that will be called as a peptide. If it is more than 50 amino acids, usually that is classed as proteins, but the general term which will cover both is basically polypeptide. Polypeptides contain peptides less than 50 amino acids and proteins more than 50 amino acids, but that is no hard and fast. This is not a sharp demarcation that we are saying. Now, again coming back to the question that if you have an amino acid which has a if you have a protein which has got a large number of amino acids, then what are you going to do? Because in one shot you can only determine up to about 30 amino acids, and if it is more than 30, if it is more than say 100, then you have to cut down the protein into smaller pieces. I told you that there are enzymes which can do that. Some enzymes are very specific. You cannot do it by treating with acid or base which hydrolyzes amide bonds. However, they are not specific. So, every amide bond will be hydrolyzed. So, you have to have some specificity in your breakage of the protein into smaller pieces. It is basically what is called tailor-made breakage. Tailor-made means the tailor cuts the piece of cloth into such smaller pieces which ultimately they tie up together and to make the dress. That is a very similar. You cut the protein into smaller pieces, determine the sequence of those smaller pieces, sequence of amino acids of those smaller pieces, and then again tie up, join them. In this joining means this is not physical joining, you again just write that what are the sequences and then finally combine these sequences to find out the correct sequence. Okay. Now, there are I also told that there are again problems with that, because if you suppose you have a protein like this, I told about that if you have cut it into say 3 pieces A, B, C, and you determine the amino acid sequence of all these 3, then the next question comes that what is the arrangement after you cut the protein into 3 pieces, then you really know that which piece is coming first, followed by which one, and what is the third one. So, what you need to do, if you have 3 pieces, then you can adapt that what is the N terminus by Sanger's method, what is the C terminus, that is by the carboxypeptidase method. And if you do that, then you know which one is actually in between A and C. Now, let us talk about some of these. What are these enzymes? That which can selectively cut molecule, the protein molecule into 3 like this. Okay. So, they are called artificial scissors, they are basically artificial scissors. Okay. Let us now talk about what are these artificial scissors or the enzymes that we are talking about, which has very special selectivity in cleaving the peptide bonds at certain positions. Okay. Now, the enzymes that are used for this, enzymes, remember enzymes are they also are proteins. So, basically proteins which have catalytic power, they are called enzymes. Now, here we are talking about enzymes. So, these enzymes, because they break the protein, the peptide bond, so they are collectively called peptidase, because they are breaking a peptide bond. Okay. Now, what are some of these enzymes? They are all naturally made, and one particular one is what is called chymotrypsin, another one is called trypsin, then you have pepsin. So, these are some of the proteases or peptidase. You can also call protease, because they are breaking the peptide bond, they are breaking the protein into parts. So, they are also called proteases, sometimes called proteases, sometimes peptidase. Okay. And this these are the enzymes which are mostly used, trypsin and chymotrypsin, because they are the ones which is extremely specific. Specific means they only recognize certain amino acids, and only when they recognize those amino acids, they will cleave the peptide bond associated with those amino acids. Okay. Any enzyme which has got a broader specificity means identifying many amino acids out of these 20, then you have the possibility of cleaving the peptide, the protein, into many fragments, which we also do not want, because we want fragments which is sizable, which has got optimum size as well as the number should not be very large. Unnecessarily, if it is cut into 10 pieces of 10 amino acids, then that does not serve the purpose, because one Edman Edman cycle ultimately will lead to the determination of about 30 amino acids. So, no point in selecting an enzyme which has got a very broad specificity. So, that is why people have selected this chymotrypsin and trypsin, because they are extremely selective, their selectivity is pretty narrow. Pretty narrow means they only recognize very specific certain minimum number of amino acids. Like chymotrypsin. Chymotrypsin, what it does, that if you have a peptide bond CONH, and if it happen to be that there is an aromatic amino acid on this side and another amino acid on this side, then chymotrypsin cleaves the peptide bond between this aromatic amino acid and the other amino acid from the C terminus end. Okay. What that means, it will cleave, chymotrypsin will cleave only when there is phenylalanine, that is one aromatic amino acid, remember those, then tyrosine, that is Y, and W, that is tryptophan. So, these are the 3 aromatic amino acids. So, if they are present, then the peptide bond will be cleaved, but remember this aromatic amino acid is attached by 2 peptide bonds, one is on the N terminus side and another is in the direction of the C terminus. So, it is breaking the peptide bond involving the aromatic amino acid at the carboxylic end, okay, at the carboxylic end. So, what I mean is basically this is also again connected to another peptide bond on both sides, left and right side. So, it actually, so what is left and right? Here you have to say aromatic amino acid, it breaks the peptide bond involving amino acid at the carboxy end. Okay. So, the C double bond O at this end, because that is the carboxy end on this side. Okay. So, that is about chymotrypsin, and but one thing you have to remember is that this amino acid, there is one restriction, that if this amino acid happens to be proline, then the hydrolysis cannot take place, that you have to remember. So, if the next amino acid which is connected to the aromatic amino acid happens to be proline, then this hydrolysis cannot take place. Okay. What about trypsin? Trypsin hydrolyzes very similarly, but it only recognizes highly basic amino acids like lysine and arginine. So, in this case you have basic amino acid. A basic amino acid means amino acids having a very highly basic side chain. We are not talking about histidine here. I told that histidine, although it is grouped into the basic amino acid, but its basicity is much less as compared to arginine and your lysine. Okay. So, if there is a basic amino acid, then also the hydrolysis will take place, but this time the enzyme that is required is trypsin. Okay. So, trypsin hydrolyzes again the amide bond at the carboxy end of a basic amino acid. On the other hand, pepsin has a much broader specificity, it actually can hydrolyze quite a few of the amino acids. So, pepsin is generally not used to determine to cut the protein into smaller pieces. Okay. Now, let us go to the next page. So, the next point, before I go into the into any other topic, let me first solve one problem that based on whatever we have learned so far. Okay. I have some pictures here which will show you that what is trypsin, how does it look like. So, this is the crystal structure, this is the crystal structure of trypsin. Okay. And this trypsin, chymotrypsin and pepsin are essential enzymes for food digestion. So, when we take a food which contains lot of proteins, so these proteins has to be broken down into smaller fragments and ultimately the amino acids, so that it is absorbed through the intestine and then they are again used to make the different proteins which we need in our body. Okay. So, first there is a catabolism. Catabolism is breaking down the large molecules. So, we take food containing proteins and these proteins are hydrolyzed with the help of trypsin, chymotrypsin and pepsin to ultimately the individual amino acids, which are then absorbed and then used by the system. Now, trypsin, it looks like this. As I said, its specificity is that it recognizes basic amino acids, lysine and arginine, strongly basic amino acids. Then you have chymotrypsin. This is chymotrypsin, that is the crystal structure of chymotrypsin. What it does? It recognizes only the aromatic amino acids. Okay. The aromatic amino acids have a big hydrophobic side chain. So, the question is why they are recognizing certain amino acids and not the others. It is basically that when we learn enzyme chemistry, we will see that what happens is the molecule goes inside and there is a big hydrophobic pocket where the aromatic groups first binds through weak interactions, hydrophobic interactions, and then the hydrolysis or the reaction goes on. In case of trypsin, there must be a pocket which is negatively charged and it recognizes basic amino acids which are positively charged at the normal pH of 7.2. So, that will go and bind there. Okay. That is why where from the specificity comes. And then you have pepsin. Pepsin is the other one. I said pepsin is little bit broader, it recognizes other peptide bonds, although here it is written only phenylalanine, tryptophan and tyrosine, that means the aromatic amino acids, but I think later on it was found that it can hydrolyze even it can recognize even other amino acids which are also hydrophobic, not only aromatic, because hydrophobic groups belong not only to the aromatic chemistry or aromatic groups, it also is present in leucine, isoleucine, valine, but these are aliphatic hydrophobic groups. So, pepsin is that its specificity is little less, that is why people do not rely on pepsin so much. Okay. Now, I told you that when you take the food, it has to be digested and fragmented into smaller the amino acids, and then these amino acids are absorbed and are used to make the proteins that we require. I think with that classification, that means the question is, can we not make our amino acids inside the body or not? That is the important, that is the question, and you know there is a classification of amino acids based on that whether we can prepare the amino acids in our body or not. And when the amino acids cannot be biosynthesized in the body, those amino acids are called essential amino acids. These essential amino acids, there are 10 essential amino acids which have to be supplied from the diet, and the rest 10 which we can prepare inside the body. Okay. So, that is a classification and possibly you know this. This is the essential amino acids cannot be formed in the body, it is essential to be taken through the diet, and the others are non-essential amino acids, they can be made in normal enough amount for in adults and children both. Okay. So, that is another type of classification of amino acids. Now, let us let us do one problem. Okay. Okay. No, before that I think let me again remember I told you that this trypsin or chymotrypsin and even other proteases, they fail to break the bond even if there is amino acid of their choice which they recognize only when there is a proline adjacent to it. Okay. When they disconnected through proline, and the reason is that proline is a secondary amino acid and it distorts the structure at that point, and so enzymes cannot accommodate the side chain of the amino acid which they prefer. That is the reason that proline stops the hydrolysis at that point. Okay. That you should remember. Now, the question is, are there any chemical method of selectively cleaving a peptide bond? The answer is yes, there is one reagent which has been developed, that is called cyanogen bromide. Cyanogen bromide is a very simple reagent, CNBr, okay, that is called cyanogen bromide. And this cyanogen bromide is very specific in breaking the bond, peptide bond associated with only methionine. Peptide bond associated with methionine. Okay. So, it only recognizes one amino acid in that sense. Okay. And what is the reaction that takes place here? The chemistry that takes place is, first of all, associated means, again you have to be very clear that if it is methionine, the code is M, and if it is NH, then this is the bond which is being cleaved, that is by cyanogen bromide. Now, let us try to draw the mechanism, or the mechanism might be given here in the slides. I can show it here. See, this is what is a peptide in the extended form, it has been shown. And where is the methionine? This is the methionine part. Methionine side chain is CH2 and then SMe, that is methionine. It is a very important amino acid. You will later learn that this methionine has a distinct role. If we cannot make methionine in our body, our protein synthesis is going to collapse, but that is not the issue right now. We are trying to see that how cyanogen bromide is able to cleave the peptide bond associated with methionine. So, this is your methionine part and this is the peptide bond. Okay. And now, as you treat cyanogen bromide, you know in cyanogen bromide this carbon is highly electrophilic, being flanked by two electronegative groups. Okay. So, it attacks the cyanogen bromide and in cyanogen it attacks the carbon and the bromine leaves. That is a good leaving group here. So, the bromine leaves, and as soon as bromine leaves, so you get a species which is like this. S methyl was already there and now S that will be attached to a cyanide and the bromide ion leaves from the cyanogen bromide. But in the process what you have created is a sulfonium ion, a sulfur which is having a plus charge. And now, what will happen as you have done this, that means this has become a very good leaving group now. What the sulfur will want now? Sulfur will want to break this bond. So, how it will happen? It will happen by any of the adjacent nucleophilic center. So, if there is a nucleophilic center nearby which can attack this carbon, and if the size of the ring is quite appropriate, that is also important. If there is a nucleophile which attacks this carbon, but that can form only a 4-membered ring or a 3-membered ring, that may not happen. In this case what happens, there is a nucleophilic center and that is basically this nitrogen and this peptide or the amide carbonyl. Okay. Now, between these two what happens, the nitrogen lone pair, as I told you last time, that this is in conjugation with the carbonyl. So, that means the oxygen is having the greater share of the negative charge between the nitrogen and the oxygen. So, you just write it in a different fashion, another conformation, that means the carbonyl on this side and this is the same peptide bond is written in another conformation, just to show you what is happening here. If you write in this fashion, then you see this oxygen is quite near to this carbon, which is now suffering the pressure from the sulfur. Sulfur wants to go away, taking the bond electrons along with it. So, what will happen now? This whole thing will be assisted by the nitrogen. Nitrogen lone pair flows it here and then the oxygen becomes negative, that attacks the carbon and this goes off. Okay. So, finally, the molecule that comes out is this CH3SCN as the byproduct. Now, question is what happens to our protein? The protein is now first it will be in this form, this iminium form, the nitrogen with a plus charge, and then this will not be very stable. Now, water comes and attacks this carbon and then ultimately hydrolyzes. This is like an imine. So, this hydrolyzes, this is hydrolyzed by water and then it ultimately leads to what? Leads to a cyclic 5-membered lactone. That was the fate of this lactone comes from the methionine. And what was the result of this? The result is that this peptide bond which we have started, that peptide is now gone. That peptide bond is not there, and you have a new peptide which is devoid of this from the left, from this left side, whatever is residing here, whatever the number of amino acids here, that is gone. So, the methionine part is converted to the N terminal peptide fragment. So, this is a very important reaction, cyanogen bromide, very specific only for methionine. However, this lactone, now this is called this is called homoserine lactone. Why it is called homoserine lactone? Because what is serine? Serine is a carbon, this alpha amino acid, and then there is a CH2 and then a OH. In this case, what is happening? There is a CH2 that is serine, then you have another CH2. So, that gives you the homomer of serine, not the homomer, sorry, the higher homologue of serine, and then you have the oxygen. So, it is basically homoserine which is converted into the lactone. So, ultimately it becomes a homoserine lactone. Okay. So, if that is formed, then you know that there is a methionine at this position, and the rest of the fragment, that is the other peptide portion from the C terminal end, that remains. Now, the cyanogen bromide reaction however, does not work for, there are 2 amino acids, it does not work if the second amino acid, if there is yes, the second amino acid happens to be a serine. Serine is CH2OH. You can see here that this is serine and this was your methionine. The first reaction takes place, no problem, the bromine goes out. So, you get this sulfonium ion. Now, the next, the peptide bond, the oxygen comes and attacks this carbon, the sulfur leaves. The chemistry is very similar to what was happening earlier, but now there is what happens, instead of, if you go just along this whole route, what you will see that ultimately you end up not as the homoserine lactone, it ends up as the homoserine itself. It ultimately ends up in the homoserine itself. Okay. So, if that happens, that means you are not breaking the peptide bond here. So, although it has reacted with cyanogen bromide, the ultimate effect is that your methionine is converted into a homoserine. That is the end product of it. Okay. This mechanism you can try by yourself, but it is very similar to the earlier one, but I leave it to you as a problem that what is the mechanism. I am not going to share the slide right now. The slide shows that how does it really ultimately going to the homoserine. So, again I repeat, if there is a serine after methionine, then what happens? The methionine becomes homoserine and that is the end product, and there is no breakage of any peptide bond. Okay. So, if there is no breakage, it will be really difficult to now tell that whether there was a methionine present or not. The reaction also fails if there is cysteine after methionine, or what happens to cysteine, because you can expect a very similar type of reaction like methionine. If there is a cysteine, what is going to happen? Will cysteine also react with cyanogen bromide and finally, it will also break the peptide bond? No, it does not. What happens with cysteine is that the cysteine will definitely react, it is SH, it reacts with the cyanogen bromide, and so it forms SCN and the bromide ion leaves. The problem is now the sulfur does not have any positive charge, because earlier it was having the hydrogen. In methionine it is Me, but in your cysteine, it is SH. So, the hydrogen is lost here. So, sulfur does not have any positive charge. So, it is not a good leaving group now, and then the ultimate result is the cyanide gets hydrolyzed. So, if the cyanide get hydrolyzed, what you get is this is this hydrocyanic acid, and what is this is there should be a CH2 in between. So, that becomes again back to your serine. So, the serine reacts with cyanogen bromide, and the cyanogen bromide ultimately is converted into the into this acid, cyanic acid. Okay. Cyanic acid is the end product, but there is no cleavage of any peptide bond. So, that is the chemical reagent that we are talking about. So, there is a chemical method of breakage. So, now we know how to cleave the protein into smaller pieces by using enzymes like trypsin or chymotrypsin, or you can use a chemical reagent if you want to know the methionine, whether methionine is present or not, or how many methionines are present. All these can be done by treating with cyanogen bromide. Okay. So, that is the how now the whole machinery works. Everything is now automated. Today the protein sequencing has become an automated. There is a machine. So, you program it and everything will be done. First it will be a digestion with trypsin or chymotrypsin, and we call it tryptic digestion or chymotryptic digestion, and then it will be separated, the pieces will be separated, and it will directly go to the amino acid analyzer and then do the Edman degradation, and ultimately the data will come out in the computer screen that these are the sequences. And finally, by the use of bioinformatics tool, you can ultimately determine whether the fragments, what is the attachment of the individual fragments. With that, you determine the what is called the primary structure of the protein. Okay. Thank you.